Bacteria and babies. What you thought you knew.
Human beings are able to break down and digest a wide variety of nutrients due to their highly versatile intestinal system. However, human babies do not have many of the necessary bacteria strains within their gastrointestinal tract necessary for proper digestion and must acquire these strains. One example of a bacteria needed for later in life is bifidobacteria, which is commonly found in the adult human intestine.
When babies are breastfed, they receive samples of bifidobacteria from their mothers, which can colonize their gastrointestinal tract and ensure healthy development. Bifidobacteria are beneficial to newborn infants by adhering to their intestinal cells. This adhesion determines the extent certain inflammatory genes are expressed in the gastrointestinal tracts of babies. The greater the amount of adhesion, the less the inflammatory genes are expressed. The less inflammation in the intestinal tract of the babies, the happier the baby.
Understanding the behavior of bifidobacteria is important when developing probiotic medication for newborn infants. It is known that bifidobacteria are anaerobic bacteria and therefore perform glycolysis and fermentation to produce ATP. Research is being done to learn more about their behavior. Through this knowledge gained by examining this species, mothers with certain chronic diseases who cannot breastfeed their infants can still be secure in the knowledge that their children will enjoy a healthy gut flora.
The bifidobacteria use organic compounds within breast milk as their carbon source. Organic molecules are molecules that contain carbon and hydrogen and may contain oxygen, nitrogen, phosphorus, and sulfur, and have a covalent bond between carbon and carbon or carbon and hydrogen. In this experiment, 2 strains of bifidobacteria were used, Bifidobacterium breve and Bifidobacterium infantis, to test which organic compound provided the best source of carbon for these bacteria.
3 different organic compounds, lactose, glucose, and HMO, were used as carbon sources in this experiment. Both strains of bacteria were grown in each of the 3 sources. The bacteria were exposed to an intestinal cell called CACO2, and the amount of adhesion between the 2 bacteria strains and the intestinal cells were measured.
DNA has all the information needed for life. All our physical traits are written out in our DNA by different sequences of the nucleotides adenine, thymine, cytosine, and guanine. DNA can be copied and made into mRNA by RNA polymerase. Transcriptional factors encourage, or in this case discourage, the building of mRNA. With a negative transcriptional factor, RNA polymerase cannot attach the DNA strand. After it is transcribed, mRNA is translated into proteins in ribosomes. Proteins determine the amount of inflammation in the intestinal tract. Inflammation can be reduced by stopping the DNA from being transcribed into mRNA. This is called negative gene regulation. The authors knew that the amount of adhesion between the bacteria and the intestinal cells influenced the extent to which the inflammatory genes were expressed. Gene regulation could be influenced by the presence or absence of different proteins, markers, or molecules. Therefore, by their reasoning, the more bifidobacteria adhesion to intestinal cells, the less the inflammatory gene was expressed, the less mRNA would be found in the cell. The amount of mRNA present could then be compared to the amount of adhesion, assessing for correlation.
The authors determined that HMO-grown B. infantis adheres to CACO2 cells much more efficiently than B. infantis grown in lactose or glucose. B. breve, on the other hand, appear to stick to CACO2 cells the same extent regardless of the carbon source used.
The authors also determined that bifidobacteria grown in HMO suppressed expression of inflammatory genes in CACO2 cells to a greater extent than when grown in lactose or glucose by measuring the amount of RNA present in those cells. This was true for both B. breve and B. infantis.
Gastrointestinal diseases such as Crohn's disease and ulcerative colitis seem to be related to the expression of the inflammatory genes in CACO2. Since HMO-grown bifidobacteria prevent expression of these same genes, The authors suggest it might be possible to treat these disorders using probiotics grown with an HMO carbon source.